Okay. Hello, everybody. Good to see you all, and welcome back to the VMOL seminar. Today we continue with the GNPS2 series, and yeah, it's awesome to have Ricardo da Silva today give the presentation on the ChemWalker, which I think is an awesome tool to expand your annotations in your molecular networks. But before you get started, just a very brief introduction about him. So Ricardo has a very interesting career path. He first did a bachelor in forestry at the Federal University of De Sousa, before he then moved on with genetics for his master's, and then transitioned more and more into the computational world, and did then his PhD in bioinformatics, followed by a visit in the Dorstein lab as a grad student, and then he also came back as a postdoc, before he then started his own independent group back at the University of São Paulo. And yeah, there in the very same theme, they I think keep doing monumental work in computational metabolomics, and yeah, I'm very much looking forward to see the presentation about one of the tools that your group is developing. Floor is yours.
Thank you, Daniel. Good morning for all of you that are listening from the US. I guess good afternoon for all us Brazilians, and maybe good night for Europeans and people on East, right? Thank you very much for this invitation. I really appreciate having this opportunity to be part of this community. It's always nice. I was telling Daniel, we are glad that we are always included. We have a series in Portuguese. It really helps groups in Brazil. I have 40 minutes, right, Daniel? If people want to stop me for questions, I'm okay with that. I don't know if you are doing that or you use to have questions after the presentation.
Totally up to you. Typically, we have like a question and answer session afterwards, but if you want to have it as interactive as possible, that's totally fine.
Okay. So I'm going to give a brief introduction on the idea of propagating annotations on networks. I believe many of you are familiar with this idea. Many of you have used NAP or are still using it. And I will use NAP as an example of propagation. And I will try to show the documentation and do a worked example with ChemWalker. If you want, you can stop me anytime. So I believe GNPS needs no introduction, but it's just to show where the concept of propagation comes from. So we are used to apply GNPS to analyze our mass spectrometry data. We usually do that by searching spectral databases and also clustering our data with spectral networks, right? So the chemists are generally used with the graph representation where they have a hit on a library and they can manually propagate the annotation for the neighbor nodes. The problem here, or not a problem, but the limitation is that the spectral libraries do not cover much of the chemical space. They are growing very fast thanks to the GNPS community for the most part, but we can still explore structural libraries. So structural libraries are orders of magnitude larger than spectral libraries. There are different ways we can use these structural libraries. One of those is using combinatorial fragmentation. The idea is very simple. If we have like a schematic representation of a structure here, we could break every bond of this structure and compute what would be the fragment we could generate breaking this bond. Then we can compare with our experimental mass spectra and see if we have matching peaks to these fragments that we generated. The problem with this approach is that generally we have multiple candidates. So here we have a representation of typical molecular networks, and nodes in red represent hits on the spectral library. Nodes in blue represent the ones we have hits on structural library using in silico fragmentation. In this example, we are working with combinatorial fragmentation. So we typically have multiple candidates. One thing that we can do as we trust more the hit from spectral library, we can use this neighbor that has a hit on spectral library to re-rank the candidates we had with in silico fragmentation. So we do that using structural similarity. So the idea here is that the candidate that will be more likely for this neighbor node will look similar to the one we have on the spectral library match. So using this idea, we propose NAP, and there are two main forms of scoring these structures. One is using a neighbor from spectral library match, and the other one is using the consensus of the candidates of nodes that are neighbors on the network, but don't have a match on the spectral library. So the idea here is that the set of best candidates for all these neighbor nodes that are connected on the spectral network will be more likely if they are similar. So basically, two ways of scoring, right? Consensus scoring and fusion scoring. NAP, it works very well. I'm going to talk about the limitations, right? It's a combinatorial experiment. We generate a lot of false positives, but if you are careful to use it, it can be very useful. But there are some limitations here. The first one is that this approach, we only looked at the direct neighbor, right? The first neighbor of the node. And the second one is that there was a very heavy application. We use the cluster where GNPS runs, but we have memory limitations and so on. So we decided to create an application that anyone can run in their own computer, and it's faster and also improves the model, and we can propagate an annotation further than the closest neighbor. This was the scoring function that we use to NAP. You don't need to remember this, but it's basically a weighted average. So we use the score given by the in silico tool. Here we use matfrag, and a match between the structural similarity and cosine score between the nodes we are comparing. So it's really a combination of on the ranking the in silico tool gives us with the information that the spectral networks also provides. And we are going to use the same idea to create a scoring system and probability for random walks on the network. Before I get to ChemWalker, I just want to browse the resources we have on NAP. I think still many users are not aware that we have some tools that orbitate around NAP that can be useful. After that, we can talk about ChemWalker itself. Feel free to stop me at any time. So this is the link that is on the presentation. I don't know if it's made available, but you can see the QR code on the video later. We have a very short tutorial here, and in this tutorial, I will call attention for two things. The first one is we have a small web server that is available to anyone. It's a very simple application that you can input smile strings for structures that you are interested, and convert it to a format that you can use either with NAP or with ChemWalker. This is very powerful because I'm going to show you some of our publications and our ongoing work. It basically allows you to search a given structure on thousands of nodes. Generally, there are lots of people that still do mass spectrometry. They calculate the exact mass and they go to Science Finder and they do this manually. So you can do that very fast and large scale. Again, you have to be careful because, I mean, just by chance, you are going to match some node on your network. But if you have a previous hypothesis of which compounds you are targeting or experiment, the protocol you use to extract a certain class of compounds, you can go to PubChem or other database, or you can even draw a structure, generate SMILE strings, generate a tabs separated file with just the structure and an ID for this compound. Then you can come to this web server. The link will be on the presentation. Don't worry about this now. And you can generate formats. For both NAP and ChemWalker, we use for the outputs of these tools, we also provide the chemical class assignment. For now, this web server can assign chemical class with NP classifier and classifier. People are loving NP classifier for natural products. They say has I don't know, it's better likelihood to be the correct class. And just call attention here that the format for NAP is PSV, is not TSV. So you just have to, for NAP is TSV and for ChemWalker is PSV, sorry. So you just have to change this format. You input your table, tab separated table with your structures, and the tool will send you an email with the result. It should be very simple to use. If you have any problems, you can email us. And the other thing is just going to go through the NAP results very quickly, just to give you an idea what this propagation gives us as result. It will be helpful when we browse ChemWalker results as well. So we have this web viewer for NAP here, just to illustrate the example. So basically you have the IDs, the mass, and retention time from your original data on your molecular networks. And after you run NAP or ChemWalker, you have, we also recover the library IDs. And for each column, as I mentioned on the presentation, we can have fusion ID or consensus ID. We just have fusion for nodes that have a spectral library match as a neighbor. If you don't have a spectral library match as a neighbor, you can only have consensus ID or MetFrag ID. So the idea of this result is basically, I want to display the results of the candidates compared with the MetFrag. So here we have a result for MetFrag on the right, and on the left we have a result from ChemWalker. Oh, for NAP, I'm sorry. So the idea of this visualization is, okay, is propagation helping my structural annotation? Because if it's helpful, you should see structures that look more similar between them. And if so, you can go further and explore the results. We can also see the structures ranked, right? There is a ranking score. I'll talk more about this scoring when I explore ChemWalker results. And you can always compare the results of your propagation with the results given by MetFrag, right? you should expect that the ranking provided by your propagation is more meaningful than the one done with the in silico fragmentation tool. Keep in mind that the main advantage here is that the propagation is taking into account your sample, right? The reactions that may be happening in the biological system that you are analyzing, while the in silico fragmentation tool is done individually in each spectrum. So it does not have context to assign annotations. The last thing I want to show is you can access the complete information for your in silico fragmentation tool. I will show you on ChemWalker that you can provide your custom database that you create with our tools, or you create in-house, or you can use one of the databases provided by either NAP or ChemWalker. And on this result, we have all the candidates, right? The first filter for the candidates is the exact match for the type of adduct that you choose. So the exact mass has to match in a certain window to the one of your node. And you have the original IDs for this candidate compound, class, assignment, and we also do something interesting on NAP that we cluster by structural similarity. So nodes, structures with the same code here are structurally similar. And one thing that we see on NAP and ChemWalker is that it's very hard to get the structure right, but the class assignment has a very good accuracy, right? We have like up to 80% accuracy on class assignment. So even if we don't get the structure right, the class assignment is generally very good, right? So this is basically what NAP did. I'm just going to show one last result on the results page. You can, let me just go back. You can go to the summary here. And if you click download here, you can download the graph of your molecular network with this candidate structures. I'm going to open it very fast here. And then I will compare with the ChemWalker result later. So this is like, we did a custom layout for NAP. So basically we have, the borders are different colors, right? We have green for spectral network match. We have blue for in silico fragmentation match here. And I think here is just one thing is that when the node, even if the node has a spectral match, it has to have the structure to enable NAP or ChemWalker to propagate the information. So I think early on, when people used to deposit data, many people did not provide the structure, right? So it does not help the propagation. So it's an incentive for you, if you are using these tools and you are going to deposit your data in public libraries, please provide the structures that they will be very helpful for the community. So here, I think we don't have the structure for this. That's why these nodes are blue. They are just consensus scoring. On the documentation, I'm not going to do it here. You can install a Cytoscape plugin that's called ChemVis, and you can do two things here. Let me just see if I have to tools. Generally, I have to first go to the settings and say that I want to do use smiles, consensus smiles, to be the where the tool retrieves the structures. Then I can right click on the node on Cytoscape. I use ChemWalker This is the Canvas plugin, and I can select paint the structure on selected node. So you can do it to all nodes. Then it can help you to analyze your data. You can also generate an SVG from Cytoscape. It will be high quality. There are a lot of people that publish images with that. You can also click on the node and go to the chemoinformatic tools and click on show structures for selected nodes. Oops. I don't know if the pop-up window is not. I don't know why it's not showing, but it should show me a picture with what?
Sometimes it's a little bit buggy. That would be actually a question I would have. So I was playing around with that feature recently, and I was wondering if you know how it scales. So does it render very complex networks completely, or is there a limitation of structures that it can draw?
I believe there is a limitation, but I mean, one of the things I wanted to say here on this presentation is that you have to have a hypothesis. It's like it does not even makes a lot of sense to draw 1,000 structures. You are not going to look at them. You are not going to produce like a visible figure with them, right?
That was exactly what I tried.
Yeah. No, you can try to do it, but it does not seem very useful. But I'm just going to show you on the browser, on the documentation, what I was trying to do here is that if you use this plugin, it generally works. I don't know what's happening here, but what you can see is that every node can have multiple candidates. So you can use also Cytoscape to see all the candidates because they are provided like comma separated on the field smiles on either NAP or ChemWalker, right? So you can have access to all these structures and see sometimes the correct candidate is not the first candidate. So you should look at least the first ones to have an idea. Okay, so any questions so far? I'm going to move back to the presentation. If you want to make questions at the end, it's okay. So this is basically the idea of annotation propagation with NAP, right? What I wanted to show, this picture was like a graph summary is not very good, but this is a publication we did with Louis, and I just wanted to call attention to this publication where we did something very interesting, right? So you explore spectral networks very well. You know that this analysis is very good to capture analogs, right? And you always know that what's clustered together is probably structure similar. Chemists love to represent structures like that, so they have side chains where they number and have many possibilities. You have to have in mind that you can simulate these possibilities and generate these structures and search in your data. So you can do it even combinatorially, right? You can have like a main group and have a list of possible connections and side groups, and then you can generate a lot of structures and search back in your data. So this will be available for you, and we did that on this publication. It was really nice. We were able to annotate a lot of analogs that were like later confirmed experimentally. So another thing that I want to call attention, we have a preprint out. It's actually a paper that no journal will accept because we did not follow the terms of service of journal. So we mined scientific literature. We downloaded like 50,000 papers, and we extracted the structures from this paper, because still in natural products literature, many people propose or elucidate a new structure, and they don't deposit this structure in a natural products database. So there's a lot of information out there that we could be using to search our spectrometric data. So this work was very nice, and this tool is available. You can do your own mining, right? You can have your literature review the papers you stored somewhere in your computer. You can mine them and extract all the structures. It's not 100%. It makes some mistakes. We have some visualization tools to help guide you on where you could edit some of the structures you capture from text mining, but it's also a possibility that you have to have in mind. You can mine structures from literature and use it to search.
So
on your own
Ricardo, what's the content of this? So you said 50,000 structures, right? So it's a structural library. And what's the structured data that's attached? Is it taxonomic or phylogenetic information? I suspect it's taxonomic information.
Sure. It's both. We mined 50,000 papers. We found more than 120,000 unique structures on these papers. Many of them were repeated over different manuscripts, and we actually built a tool you can use to mine your own collection. Okay, so this tool it's available. It's on our GitHub page, and this preprint explains how we did it, how we validated the tool, and we also make the structures that we mine available. Does that answer your question?
Yeah, so what it is it's a structural library that is created and connected to taxonomy. I don't think phylogeny, but taxonomy. Is that right?
It is actually a tool that enables you to create your own structural library.
Okay.
Okay. We did not create a library itself. We just created a tool that enables a single user to create their own library.
Very cool. So if you're interested, let's say in Streptomyces, you can search for Streptomyces and it will provide all that.
I mean, you have to provide PDF files to the tool. So let's say you are interested in Streptomyces, you go to Google Scholar, you find all the papers that have you download the ones you can, and then you use this tool to extract the structures from the papers.
Nice. That's really cool.
Yeah, I mean,
so Ricardo, right? I can ask now, taking advantage that Peter asked.
Yeah.
Because then when I the way you describe it then is let's say you have the PDF, you have the structure draw in the PDF. You have no smiles, you have nothing there, and you just grab as an image and convert that image into smiles.
Yes,
and if you have in the supplemental material, then you have the structures like in a tables. In tables, you also can grab that.
Yes, you have to provide the file, right?
You have to provide the file, of course.
That is so awesome.
Okay, that makes sense now. Cool.
Yeah, I did not. I just want to make clear there are a lot of software for optical recognition, right? We use one package that it's very it's called OSRA, and it's used on chemoinformatics. There's a lot of new tools coming out now for optical recognition, and we can even combine them in the tool later. But the tool is very simple. It's a Python package that wraps the text mining and optical recognition for the structures that are provided as figures. But you can also search mentions of compound name on the text, and it translates it to inchi or smile. it is okay?
Yeah, very
Any more
cool.
questions? Yeah, take a look. If you have questions, we'll be happy to answer and work together. We want to improve this tool and make it publishable. Okay, so just coming back to where we stopped, we have our network. We are able to do propagations in an automated fashion, but we have these limitations. You cannot use it in your computer, and you can only propagate to the closest node to your spectral library match. So with ChemWalker, we just try to reframe the problem in a different way. So for instance, if you have like these 3 nodes in your spectral network and you have candidates for each node, we can now rewrite the network with the same topology of a spectral network, but now for the candidates of each node. So it's a network of the candidates of each node, and then we can weight these new networks with the structural similarity as the edges now, and we can rewrite the same function we use to provide the scoring for NAP now to create these weights and walk on the graph. So the paths on the graph will be more probable as the weight grows on the edges. So you'll be here, I would say that it will be more probable that I walk from candidate C2 to candidate B2 to candidate A2. So these will be the most probable candidates for the network, right? So that's the idea of random walks on graphs that we use to assign candidates for the networks. So this is the random walk algorithm. 2 PhD students were involved in this project. It's published already, and now the greatest thing, it's available on GNPS2. I'm very happy with this. Thanks to Ming and Alberto that helped to implement on GNPS2, and now you can use it on web interface of GNPS2. I will show you very quickly how to do that, and you can also use it as a Python package. I will also show this now. So this is the interface from GNPS2. I will show it very quickly. And the last idea I want to show here is that we are using an approach that is called random walk with restarts. That means that we are always restarting from some nodes, and then the nodes we are restarting that are the spectral library match. So here we have in blue 2 spectral library match to this molecular family or connected component, and we always restart the random walk from these seed nodes. And what I wanted to represent here is that the green borders are the nodes that have the ranking of the correct candidate improved by the random walk from these seed candidates, and the red one is the one that has the ranking of the correct candidate worsened by this walk. So it in average, it improves the ranking of the correct candidate on your networks. It has the same results of NAP. The results are not statistically different from NAP. So it's an improvement because now we can propagate information on molecular networks for nodes that are not directly connected to the spectral library match. So that's the main idea of random walks for propagation here. I'm going to do the same thing. I'm now going to just show very quickly GNPS2 interface, and the very nice thing that you can connect directly from your feature-based molecular networks on GNPS2. So let me show you very quickly. I separated an example to show here. And we also have a documentation, right? You can access later and see the details. So I'm opening a feature-based molecular network job here, And then we have this very nice feature that we can do downstream analysis with ChemWalker here, this yellow button here. But first, I want to just to show you something very quickly. For now in GNPS2, we are operating at molecular family level, right? We are only able to propagate inside molecular families. We cannot propagate the entire network. And remember, even when you do that with NAP, or if you do that with ChemWalker running or Python package, the propagation is contained inside molecular families or connected components, right? You have to have an edge between nodes to be able to propagate the information. So if I search on this, let's say you have a node that you are interested on your molecular network, and you can see from this panel here, you can see the connected component index, right? Knowing this connected component, let's say you are interested in some node on this molecular family. So you are going to use this component index to propagate the information on this molecular family. So you have to be able to identify this component index, and then you can go back on your job page, and you can do downstream analysis with ChemWalker, and then it will be very easy to fill the form here. So just to call attention, what the redirecting that this button does, it pastes here the ID of your molecular network job, but you could also paste it yourself, the task ID, and you can even do this propagation with jobs from GNPS1, right, on feature-based molecular network version or version 2. And here you have to provide the connected component index, and you just have few parameters. You have to provide the ionization mode, positive or negative, and you have to choose the adduct type, right? Here it's mixed, right? You have to be careful because the adducts for positive mode and negative mode are mixed. Maybe we can separate them later. And we have a ppm tolerance. So this is the first filter, right? We are going to filter the structured database provided by this ppm tolerance for each of your candidate nodes on the network. So the first thing is every node in the network we search on the structured database with this tolerance window that you have to adjust to the accuracy of your instrument. We have as default a Coconut database. It has over 400,000 structures, but as I showed before, you can create your own database. See, this is the format that ChemWalker uses, is a PSV file, and here we even have a link to the documentation where we show how you can format your own database. So after selecting these parameters, you just submit the workflow, and I'm just going to show you the result very quickly. I have a result prepared here. So the result page it's very simple. We still don't have an app to analyze online, but you can download the TSV with all the candidates and also the GraphML. I'm just going to show you very quickly the TSV and the GraphML, and then we can go to the discussion section. So on TSV, we have the scoring that is provided by MatFrag, the inchi and the smiles. We also export some results of the MatFrag for the masses of the fragments, and the smiles of the fragments. The prediction of fragments is not good by MatFrag, but the masses have some meaning, right? And I'm just going to call attention from the last columns. What we have here is, you can see that for every cluster, for each cluster index on your network, you have multiple possible candidates, right? So you have the ID of the possible candidates, and you have the scoring from ChemWalker. Just a quick heads up for this scoring system here. It does not have meaning for an absolute value. Let's say a score higher than 0.8, it's good enough. We are not able to do that. We are only able to do comparatively. I can only say that a score of 0.8 is much better than a score of 0.3. It does not have an absolute value meaning here. Okay, so that's it. You have the structures of all candidates and the scoring for all candidates here. And the other result, it's a graph with the information. Let me just GraphML file. So here we have just that connected component, and this as the same for NAP. We have the structures for all candidates here. You can draw the structures on the nodes with canvas, and you can also see the candidates of structure as I pointed to the manual, and it's really easy to use. I wanted to show the result from NAP here. Is that you can follow the inspiration of this layout that we created for NAP and created for ChemWalker result. It's very easy to do that on Cytoscape, and you can just my timer just told me I have to stop talking, so I'm going to stop talking. But that's about what I wanted to discuss here.
Ricardo, you're still fine. We have the full hour till 9. So
really?
you know, then that just takes away from like the discussion session. But feel free to continue and talk as long as you want.
I'm almost done. Like I said, I just wanted to call attention for these steps here, these tools orbiting around NAP and ChemWalker. So I started telling that we can build our own structured databases to search for mass spectrometry data. And after we have the structures, we also can do downstream statistics, right? So a question that everybody is interested is like, how can I summarize the results obtained for large-scale annotation? So everybody is using chemical taxonomies, right? Classifier, NP classifier I showed before. But generally, we have still very large and complex networks to interpret, right? So here we have a representation of molecular network, color code by chemical class, and we are working now on automating class enrichment for these networks. So we have this tool PyClassRich that we are developing. I'm going to show very quickly a paper that we published with Neha, where we applied this enrichment. It's based on ChemRich, that was a web server published by Oliver Finn that provides class enrichment for GCMS. We basically use the same technique, but now we automated this, grabbing the results directly from NAP and GNPS and providing the class enrichment. So very briefly, here we can have the larger the circles, bigger the number of compounds annotated on this class in your network, higher on y-axis, more significant is your enrichment, and if it's color, the more red your node is, higher the number of compounds on the treatment compared to your controls, and more blue, the higher the number of compounds on your controls compared with your treatment. So you can basically summarize annotations for large networks with this class enrichment strategy. So, I mean, that was basically it. I want to leave some time for questions, then we can discuss. And if you want, I can go back on some of these results. This is one thing that was not done before, but we think it's maybe very useful. We can also overlay these ontologies on graphs, right? So basically, as classifier has a hierarchy classification, right? So you have superclass, then you have classes, and then you have a subclass of compounds. If we use the same idea of enrichment, we can say, okay, you have a superclass of chemical classification, but sometimes it's not very informative. So this superclass of lipids here is saying that there is more lipids enriched on your treatment compared with your control. But then if you break it down, which lipids, and then you can see that there are some lipids that are higher on your treatment, but there are some lipids that are depleted on your treatment. So using this ontology graph may help you understand what's happening on your metabolism as well. So this is coming up, I hope, sometime soon. And that's it. Thank you very much for this opportunity. And I want to thank our lab, very young, very motivated students, and the University of São Paulo as well, our research group, our funding agencies, and I will be happy to take questions.
All right. Well, thanks so much for that seminar. That was super exciting. It's good to see what you're up to these days too. So yeah, for anyone who has to leave now, we'll end the recording and then we'll take some questions.